right, so we're just waiting for folks to make their way into the active seminar room from the waiting room. So we'll give a few minutes before we get started. All right, well, as we wait for the last few folks to join here before 5 after, I'll just give us a quick introduction. So I want to welcome everybody here today on behalf of the Biomaker Space and the Biomakers Group. We have another one of our COVID-19 Biomaking Solutions seminar series. So a couple quick notes, as most of you who have attended these are familiar with, we hold questions until the end. At which point you can use the raise hand icon on the participants tab. I'll bring you off mute and you can ask your question directly. Otherwise, just like to remind you, we have a couple of more sessions coming up later in this month and in August. You can see those on the website and register for them on the website, as well as the reminder emails that come through various email lists that you may be on. So with that, I'd like to hand it over to Professor Angela Belcher to introduce our speaker.
Great. Well, thank you. Welcome, everyone. And I'm extremely excited to introduce our seminar speaker, Professor Mark Bathe, who's a professor in biological engineering at MIT. He has earned his undergraduate and graduate degrees at MIT, and he's been very involved in service as well. He also did his postdoc in Munich. He's been very involved in service, as I said. He's won mentorship awards and is involved in very innovative teaching in a program called NEAT in undergraduate hands-on learning experiences in engineering. And he has, he's currently the chair of the Committee on Student Life. His work, I think, is exciting and fascinating, and he's going to tell us a little bit about one particular topic today. But what I love about his work is it really uses the beauty of structure and self-assembly in nucleic acid systems and does prediction and modeling about how these structures can be controlled at the nanoscale and even how they can come together at the larger scale, and how they can affect applications. And the applications he's worked on are a range of applications, anywhere from new computing and memory to light harvesting and therapeutics. And so it's really, to me, it's beautiful because it's using the fundamental structures of life to have an impact in many technologies that affect humanity and society. And I think if you look at Mark's work, that really is based on things that can affect society and humanity. So with that in mind, today he's going to tell us about his very new work on making virus-like DNA particles for vaccine development for COVID-19. So I'll turn it over to Mark, and thank you for speaking to us today.
Thank you, Angie and Justin, for the invitation and the kind introduction, and welcome everyone to this virtual seminar. As Angie mentioned, our lab in the Department of Biological Engineering is applying a relatively early stage technology that's called nucleic acid nanotechnology to a diversity of different areas, ranging from computation and sensing to structural biology, and also therapeutics and vaccines that will be the subject of my talk today. So if we look across the globe at diseases and disorders that affect humans, there's really a pressing need for new gene therapeutic and vaccine technologies. Thanks to next generation sequencing efforts from the Broad and other leaders worldwide, we now know there are over 6,000 genetic disorders. If we look at cancer, just in 2019 alone, there were over 500,000 deaths just in the US. And of course, turning to the current pandemic that we're living in, there are over 500,000 worldwide deaths from COVID-19 to date. But we also shouldn't forget another ongoing pandemic that has wrought tremendous havoc for decades now without solutions for vaccines or cures, stemming from HIV, of course, with over 30 million worldwide deaths to date. And so in the therapeutic and vaccine area of our lab, we're interested in developing a new early stage technology that is leveraging programmable nucleic acids for next generation gene therapeutic and delivery and vaccines. And the reason these materials are interesting in these areas, especially as we transition from a sort of era of therapeutics and drugs that were largely dominated by small molecules and antibodies towards new drugs and modalities of drugs that include messenger RNA, interfering RNAs, antisense oligonucleotides, as well as CRISPR, is that these materials that we can program into virus-like structures are very naturally amenable to complexing and delivering these therapeutic payloads. And in addition, their virus-like structure allows us to display proteins and peptides. This is showing the trimer from GP120 from HIV on the left, in orange, displayed on with 30 copies on an icosahedral DNA nanoparticle. And so generally these particles are highly programmable on the 10 to 100 nanometer scale, which is the scale of viruses. They're relatively inexpensive to manufacture compared, for example, with some viral vectors that are being used for gene therapeutics. They enable active targeting and immune cell stimulation through the display of either proteins, peptides, or other small molecules from their surfaces. This is showing a messenger RNA that's folded into a discrete virus-like particle. The messenger RNA is in red, and it's hybridized to DNA oligos that are shown in gray that offer us then the ability to target this moiety. Alternatively, the messenger RNA can be put inside tetrahedral DNA origami and then targeted using that modality. And then finally, in the CRISPR and siRNA case, one can complex stoichiometrically controlled numbers of siRNAs to the interior of the particle, as well as the CRISPR ribonuclear protein complex, the RNP, together with its guide RNAs, and also integrate an HDR template into this nanoparticle. So as you can see, there are many different modalities, and one can also build in triggered cargo release of these therapeutics and also leverage their properties that they're made from natural materials. They're made from DNA and RNA that are also biodegradable and controllable in their degradation rate and immunogenicity. And so these different modalities in this overall area is actually online just today in a review from Marina Dobrovolskaya and myself in Wire's Nanomed Nanobiotech. So today I'm going to focus on our vaccine work, and I am not an immunologist by training. I am a technologist. I am very happy and we're very grateful to be able to work with leading local immunologists, both in our own department, Daryl Irvine, who we collaborate with on our HIV vaccine area, And then in a newer collaboration with Aaron Schmidt and Daniel Lingwood on the SARS-CoV-2 vaccine application, which I'll be showing towards the end of my talk. And I'll be also spending quite some time on the HIV work that was published recently in Nature Nanotechnology. So we're all here today, both virtually online through Zoom, and also rested in our jobs and lives and educational activities worldwide due to the current pandemic, of course, which I already noted had over 500,000 global deaths. And Dr. Anthony Fauci recently noted that this number might rise to the millions before we have this pandemic under control, with over 13 million confirmed cases worldwide and almost 3.5 million in the US alone. And so tremendous show of force of technology in the vaccine landscape to try to handle this pandemic, as well as in the therapeutic landscape, of course, in the development of neutralizing antibodies and also small molecules. There are over 160 vaccines that are already in development, and these are all over the news that I'm sure everyone is reading about and hearing about for SARS-CoV-2. And so I think one natural question one can ask is, why do we need more? Well, the fact that there are over 160 vaccines in development means that there's no clear winner. If we knew which one was going to win, we would focus on that one and develop it and deliver and have our job done. And so the idea is to really take as many shots on goal as possible, because this is such an important developmental feat, and also tremendous uncertainty in the outcomes of the various vaccine modalities that are being developed. There's been, of course, some very promising results that are in the news, including from one of our former speakers, Moderna, locally in our neighborhood here, that look very promising and are moving on to phase 3 efficacy trials after passing safety trials. But a question going forward is going to be, even if these vaccines that are in early stages develop or elicit humoral antibody response, typically the antibodies that are being elicited are neutralizing antibodies that target the spike or S protein of the coronavirus, SARS-CoV-2. The efficacy trials typically only look at response within a few months, and the question will be how long-standing that humoral response and immunity will actually be following vaccination. And of course, people have also heard about stories of patients getting reinfected, and so there's some question about how to really make vaccines that have longevity, as we're used to in some of the conventional vaccines that we already get, licensed vaccines. And more broadly in the vaccine space, we're also humbled by the challenges that we faced in developing a successful HIV vaccine. So if one looks back at the timeline of the AIDS epidemic or pandemic, it was in 1984 that HIV was identified as the cause of AIDS. In 1998, it took some years, but the first vaccine trials began. And it was about 6 years later then, in 2004, where vaccine candidates failed in phase 3 trials. And it was only about 10 years ago that scientists identified potent antibodies, VRC01 and VRC02, that were from living patients that neutralized most strains of HIV. And of course, in the case of SARS-CoV-2, this moved much, much faster. We already have these candidate antibodies in a number of different companies being in production and testing for transfusion into patients for treatments. But nevertheless, this then also focused vaccine efforts towards eliciting the production of those neutralizing antibodies, where in 2016, these antibodies were both moved into trials for testing as intravenous infusions, but also vaccine studies have been aimed at eliciting or inducing humoral immunity through the production of these antibodies. So across the different formulations of vaccines that have been developed, which will lead to a description of the approach that we're taking. There are a number of different categories of vaccines. So 4 broad categories: genetic vaccines, which are the vaccines from Moderna, Translate Bio, CureVac. They're messenger RNA and in some cases encapsulated in lipid nanoparticles for delivery and uptake, and then induction of or production of the proteins of interest, the anagenic proteins, for example, the spike protein, to elicit an immune response, or from Inovio, another company, using a DNA version that also induces production of that anagenic protein. Viral vector vaccines, the vaccine from Oxford that is also in the news and looking promising, utilizes adenovirus that's responsible for the common cold, a variant of that to similarly produce anagenic protein for immune response. And then the second 2 categories are either a whole attenuated virus vaccine, which takes a genetically modified version of the vaccine and debilitates it so it can't reproduce, but it has intact its structure and coat proteins that is also very stable and safe, and is used to elicit immune response. And then protein subunit vaccines, which use protein subunits in various formulations, including presenting them in a virus-like organization as the vaccine particle of interest. And so if one looks at FDA-approved and licensed vaccines, they largely consist of the latter 2 categories, which have been around longer for the most part. So either a whole attenuated virus vaccines, a long list of familiar vaccines that we're familiar with, and then protein subunit vaccines as well, shown on the left. And there's 2 that I've bolded that have been shown to be very effective virus-like protein formulations. So these for hepatitis B and HPV take the anagenic protein of interest and self-assemble it into a virus-like protein structure that then is used as the vaccine particle. And so these virus-like protein particles and whole particles or attenuated viruses are known to traffic well to secondary lymphoid organs, lymph nodes in particular, due to their size and stability. And the repetitive presentation of antigen on the surface of these viral particles that once they reach lymph nodes are displayed by dendritic cells as an intact viral particle antigen that primary B cells then will recognize through BCR, B cell receptor, which is coming from a large pool of diverse B cells that have different affinities for all sorts of antigens. And in this recognition step, there's clustering of BCRs that occurs, or cross-linking that induces B cell activation as these cells then will bind that native intact antigen particle, but also ingest it, process it through MHC2, and present peptide fragments to T helper cells in a second selection step that then gives them the ability to migrate into germinal centers and proliferate and mutate in this affinity maturation process shown on the right, where diversity in these antibodies or BCR is developed. And if the diversity results in a lower affinity variant of BCR, then the cell is programmed to die or instructed to die through apoptosis shown on the right. but follicular dendritic cells that are presenting this antigen within these germinal centers will also offer the new variants of BCR and B cells to bind these antigens again and be selected if they bind tightly and well on a pool of antigens that's diminishing over time. And they'll get selected and either re-enter for further affinity maturation, eliciting T cell help, or result in long-lasting plasma cells that will then produce this antibody, which is really the protective humoral response of interest, that these cells can live for many, many years and decades in bone marrow and produce these antibodies. And also memory B cells can be produced that are sort of a backup system that if they re-encounter the antigen, they will be activated as well. And so in the viral-like protein category, some antigens self-assemble on their own and some don't. And this is a very nice paper from Gary Nabel's lab at the NIH Vaccine Center showing when antigens don't self-assemble on their own, as is showing the hemagglutinin trimer from H1N1. If they don't self-assemble on their own, then one can use ferritin, which is a nanocage, to self-assemble this protein into a virus-like nanoparticle, where ferritin has a copy number of 24 and presents 24 copies of this antigen and can be fused to ferritin to genetically express this entire construct to self-assemble and produce this virus-like nanoparticle. So David Baker and Neil King's labs have taken this strategy and engineered synthetic virus-like particles that aren't limited to this copy number of 24, or we can make many more copies of antigen and also mix multiple types of antigens since 2013. But they still have several limitations that include limited control over the copy number of antigens, the spacing between the antigens, and the number of antigen types. And they also have a possibility of generating neutralizing antibodies that may be targeted to non-antigenic regions that may not allow for boosting of this vaccine in clinical use. And finally, the glycosylation that can be important for these antigens may also be non-native to the antigen. So another variant of the virus-like nanoparticle formulation that has been developed by William Schieff's lab at Scripps Research Institute used a bacterial protein that was conjugated to an HIV immunogen called engineered outer domain of germline targeting 8 that is engineered from the GP120, the CD4 binding site on this antigen. And the goal in this was to self-assemble a potent nanoparticle vaccine displaying 60 copies of this antigen that targets germline cells that can then basically develop B-cell clones that will allow for subsequent clonal selection towards generating VRC01 class antibodies of interest for a successful HIV vaccine. But using this technology of templated proteins or self-assembling proteins, there are a number of limitations that one can't quite easily surpass. And so examples of that include what is the optimal number of antigens to present in the particle, what's the spacing that maximally triggers or activates B cells in these particles, how does antigen affinity affect these parameters, and also what's the impact of scaffold rigidity itself, in other words, maintaining the distance between these antigens on B cell activation. And so engineering nanoparticles from DNA that offer the ability to ask these questions allows us to elicit or elucidate the sort of design rules for obtaining maximal B cell activation from a vaccine nanoparticle. And so this technology and approach dates back to the early 80s. So Ned Seaman, who was actually a postdoc in Alexander Rich's lab in the Department of Biology at MIT in the 70s, was working on crystallography at the time, came up with the concept that one could use sticky-ended cohesion of specific Watson-Crick base pairing in DNA together with holiday junctions that are programmed using synthetic DNA to cross-link DNA nanoparticles, or DNA duplexes, I should say, allows one to make custom nanometer scale assemblies. And so Paul Rothermund in 2006, quite a few years later, took these concepts from Ned's pioneering work and came up with a transformation in the ability to engineer nanoscale assemblies from DNA, where he used a long single-stranded DNA scaffold from M13 phage that self-assemble under thermal annealing together with helper oligos to form discrete shapes that could be controlled, shown here in 2D, and William Shee's lab extended that to 3D as well. This is just a little animation showing how these particles come together as you thermally anneal them, and you're self-assembling them with Watson-Crick base pairing to form that final shape. In our lab, we've generalized this approach of DNA origami to be able to produce any virus-like nanoparticle on the 10 to 100 nanometer scale in a top-down fabrication or design approach. And the way this works is to take the target virus-like particle shape and route the single-stranded DNA scaffold, shown in blue, throughout the entire structure, and then design the sequences of oligos that offer the ability to conjugate proteins or peptides and other small molecules, as well as antigens, through either hybridization to single-stranded DNA that is displayed from 3' and 5' ends of these duplexes, or through alternatively covalent chemistries that are available. And we can also produce these particles at preclinical scale using a bioproduction and fermentation process where we can design a custom length and sequence scaffold of the single-stranded DNA to produce with a helper plasmid approach that Andrew Bradbury originally introduced for phage. So when one produces these virus-like particles, one gets very well-formed individual particles, shown here with atomic force microscopy, that resemble viruses, only these are pentagonal bipyramids and programmed from DNA. One can also look at high-resolution cryo-electron microscopy and see that the 3D structure of the DNA assembly is matching what was predicted from the sequence design that was used. And so turning to applying this then to vaccine nanoparticles in vitro, what we're able to do is focus on two different nanoparticle shapes. One that's an icosahedron, shown here, and the yellow is showing the EOGT8 antigen from Bill Sheaf's lab, where Daryl Urban's lab helped come up with an innovative way of attaching these in a conjugation step using peptide nucleic acids that hybridize to displayed single-stranded DNA oligos, and also a simple rod where we looked at just linear presentation of antigens to ask questions about how the spacing between these antigens impacted B-cell activation. And so the goal was to take sort of the 60-mer single particle and ask the question if we can vary the copy number and spacing between these antigens and these different constructs. What can we learn about how B cells recognize vaccine particles, and then can we use that to design better vaccines ultimately? So the first observation in a B cell reporter cell line that's from Daniel Lingwood's lab. This is a Ramos B cell line that expresses VRC01, which is the target receptor for this icosahedral particle displaying the EODGT8 where the fluorescence indicates on the vertical axis the degree of calcium signaling, and over time on the horizontal axis. And you can see for either 0 or 1 copy of the antigen, one gets no activation, no recognition. With 2 copies, one gets some activation in blue. And the surprising finding was that with only 5 copies distributed around the particle, one got similar levels of activation as with the intact 60 MER protein nanoparticle. And that's quantified on the right as well, where you see what's a monotonic increase in the degree of activation or signaling as the copy number of the antigen is increased to 5 and then 10 compared to the positive control 60 MER nanoparticle. We then turn to the question of spacing between antigens, which has not been elucidated before in B-cell activation, what its impact is, and looked at a rigid rod presenting just a dimer of antigens. And what was interesting was that as the spacing between the antigens was increased from 7 to 14 and 21 nanometers, and then finally 30 nanometers and even 80, there was a monotonic increase in B-cell activation out to about 30 nanometers, and then a plateau, which goes counter to some of the thinking in the field that maximally packing antigens as densely as possible on a particle leads to the best triggering. We were able to turn to the icosahedral particle and saw a similar dependence on spacing, where again there was this monotonic increase as one moved from tightly packed 3 nanometer spacing between the EOD-GT8 out to 11, 15, and 22 nanometers. Another question in the field has been, what is the impact of the rigidity in terms of the spacing between the antigens on B-cell activation? And so in this case, the lead authors, Remy Veneziano and Tyson Moyer from Daryl's lab, took the dimers and asked the question, well, if instead of being presented on a DNA origami rigid fixed spacing, if I instead present them on PEG linkers or single-stranded DNA linkers that are flexible, what happens to B-cell activation and signaling? And what we saw was a significant decrease in activation as one moved from a rigid backbone scaffold to a flexible polymer scaffold. And on virus particles, of course, that are used in some of the formulations I showed, the protein virus-like particles, they also have that fixed spacing. It's in contrast to a fluid membrane, where we compared with Daryl's lab also, who did all of the cell-based assays here in vitro, comparing a DNA origami presenting a fixed copy number and spacing of antigens on the particle compared to a liposomal formulation, shown here, lipos, with a peptide from the Irvin lab that had 2 different affinities, so P5 and P31, or a medium and high affinity peptide sequence. For this now in primary B cells. And what you see is that the red and the purple curves are the pentagonal bipyramid origami construct, where the initial activation is significantly higher than the equivalent density or copy number of antigens presented on a fluid membrane, where the antigen spacing can change due to diffusion within the membrane, corroborating also the result from the one-dimensional presentation. Matthew Stone in our lab then turned to look at B-cell activation downstream of origami by doing confocal imaging, by adding the antigens and then imaging the EOD GT8 itself, which is on the nanoparticle, 30 copies in this upper panel shown here at time 0, where it's unstimulated. And these are fixed time points then, where BCR, the B-cell receptor, is stained in green, and phalloidin, which stains actin in the cortical matrix that's below the cell periphery, in blue. And after 1 minute, you can see punctate red dots forming, where the viral-like nanoparticle has engaged BCR and bound, followed by endocytosis and uptake in 5 minutes and 3 minutes. With at 5 minutes, you see a significant enhancement of also the cortical region, together with the co-localization of BCR with the EOD antigen. Matthew was able to also then quantify downstream signaling, looking at SYK as well as Lin kinase, which are downstream of B cell receptor activation, and showed that the 30-mer, for example, had similar levels of activation or signaling compared to the EOD GT8 60-mer, which corroborated the calcium flux measurements that were shown previously. So some of the earliest models of B cell cross-linking or BCR cross-linking and activation was provided by Dinsis et al. In a PNAS paper all the way back in 1976, where they used a flexible polymer and varied the number of antigens to in vivo look at antibody production, and basically came up with what was called an immunon model, where they postulated that a specific number of antigens that are on the polymer at the right spacing needed to bind individual B cell receptors and cross-link them up to form a discrete complex that they call the immunon. Our data basically build on this model by suggesting that the activation monotonically increases with the number of immunogens, but also not only with the copy number of antigens, but also with the spacing between the antigens up to about 25 to 30 nanometers. And that the fixed rigid spacing between these immunogens is essential for robust B cell activation. And so with collaborators at Ohio State University, Children's Hospital, Professor Jayajit Das, who's a computational modeler, who models how BCRs in cell membranes interact and diffuse, and also signal downstream with these kinases that I mentioned, Lin and SYK, was able to come up with a relatively simple model that explained our results of a monotonic increase in activation as the spacing D between antigens is increased. And what it postulates is that these nanoparticles basically bind to B cell receptors in small microclusters or nanoclusters. And if they're too close apart, there's significant overlap of those clusters that reduces the overall copy number of BCR and downstream signaling molecules in those domains. Whereas as you start to separate apart these antigens, you basically separate those microclusters and allow for decrease in sort of steric occlusion that reduces otherwise the copy number of associated BCRs and signaling molecules until these microclusters are separated away, and then this plateau is reached. This is just one very simple possible model. There are many other models, and this is a very big field of investigation, or certainly not claiming that this is the only explanation, but rather one simple one that might offer an explanation into the observations that we've had. So because the technology we're using is a platform technology where one can swap out the antigen of interest very easily, we were able to pivot this HIV vaccine platform to SARS-CoV-2 together with Drs. Aaron Schmidt and Daniel Lingwood at the Ragon Institute and Harvard Med School. So Aaron Schmidt's lab modified the RBD, receptor binding domain of the spike protein that engages the ACE2 receptor on human cells to infect them, and has been the primary antigenic target of interest to generate neutralizing antibodies. They were able to do this in only a couple of weeks. And in our lab, then Dr. Eike Womhoff and Grant Knapp were able to, in several weeks, also produce nanoparticles displaying the RBD from the spike protein. And what we're in the process of is generating libraries of these particles where we can vary whether they're presented in trimers or spaced equally throughout the particle in varying copy numbers and also sizes of nanoparticle, and evaluate which of these act as optimal vaccine nanoparticles, both in vitro and also in vivo. Most importantly. And so this is just showing some first preliminary characterization results that show from Aaron's lab that the nanoparticles bind similarly to the wild type receptor binding domain, the RBD. This is just basically expressing ACE2 receptors in a HEC cell line. And what you can see in the shift is that the nanoparticles are able to bind. This is reported with the CR3022 antibody that binds a different neutralization site than the ACE2 binding site. And so that looks promising. So going forward, I should have said at the outset that I was going to present mostly HIV work and in vitro characterization, so that I didn't disappoint too much. But all the mouse studies with these works, both in Daryl's lab and also in Daniel's lab, are ongoing. And I don't have anything to share today, but hopefully I've given you fodder for thought about how we can take antigens on these DNA nanoparticles and control their spacing and copy number and size of the particle to elucidate in vitro basic rules of vaccine design that, at least for isolated B cells, tell us what maximally signals or triggers them. And of course, as we go in vivo, there are many very important open questions and challenges about how they traffic to lymph nodes, how stable they are, how T cell help is induced, and signaling downstream in affinity maturation. So that's what I wanted to share with everyone today, and see we have a couple of questions or comments already in the board. But hopefully I've convinced you that these DNA virus-like particles offer a versatile rapid candidate vaccine platform for different pathogens, including HIV-1 and SARS-CoV-2. That they allowed us to, for the first time, elucidate some basic design rules for B cell activation that remain to be tested in vivo, and also with, for example, the RBD from SARS-CoV-2. And then these ongoing in vivo studies will importantly be, of course, led by the expert immunologists, Daryl and Daniel Lingwood and Aaron Schmidt, as we provide the materials that evaluate the role of, first of all, B cell signaling through TLR9 that will recognize the DNA-based material that our particles are made from as they get endocytosed, their long-term stability in germinal centers, and then, as I mentioned, the role of T cell help in this affinity maturation process that Daryl's lab is really expert in. All right, with that, I'd like to thank everyone for joining, and again, thank first-rate collaborators that we're very fortunate to be working with as we, as engineers, are developing this early-stage material for exploration in this complex space of vaccines and therapeutics. And I want to thank Remy Veneziano, who was the postdoc in my lab, who now has his own lab at George Mason University, who really did the brunt of a lot of the work that I showed today. And also Eike Womhoff and Grant Knapp, who have been pushing hard on the SARS-CoV-2 project to make progress with that as well, and as our funding sponsors. And thanks again, Justin and Angie, for the opportunity to share our work with you today.
Thanks, Mark. So as you mentioned, we have a couple of questions up in the chat already. Robert, would you like to ask yours out loud? I can bring you off mute if you'd like to do that, if you are still in the session. All right, not seeing any response there. Mark,
Yeah,
maybe.
I sort of got the gist of it, and I think it's a great question. So we are using DNA to make our nanoparticles, and I think or RNA in different cases, and I think he's asking maybe how this might be potentially toxic or dangerous for recombination that might get incorporated into either the virus or our own cells. And I think maybe I think it's a great question, great thought. The idea is that our DNA sequence is fully controlled by us, so we can make junk DNA that's not protein coding, has no promoters, has no function. On the other hand, it is still DNA based, so there is some risk of that by providing that material, but there's a lot of DNA around anyways from apoptotic cells and other cells as well. So. But Bob is, of course, a leading expert, so it'd be great to follow up with him on the biology of these applications of these materials, since I'm, I think Maxine raised her hand.
Looked like it. So folks who have questions, who are curious, just as a reminder on how you can use the raise hand feature under the participants tab, click next to your name, you can click the raise tag, and that enables me to bring you off mute. And you can ask the question directly. Oh, there we go. We got Christopher here. Chris, here we go. You should be able to unmute and talk.
And I can also read. Actually, there's some questions in the chat that I could also read if.
Maybe we'll alternate. So let's try taking the one from Chris.
Okay. I think he might be muted there. I just heard a little bit of sound come through.
Muted now. All right, maybe let's we'll switch over and take one from the chat. Or
okay.
Wait, Chris, maybe off mute now. You want to try real quick?
So Preeti Krishnamani asked how long the particles remain active in the body, and so that's something we've been very keen to understand, but unfortunately the in vivo studies in mouse ongoing in Daryl's lab and booting up in Daniel's lab are still active, and so we don't have yet very good answers to that question, or I should say any. What I can say is that these materials, we're not the first ones using these materials in vivo, and a number of other groups have done so, and the unmodified DNA versions of these particles degrade due to DNase I relatively quickly, and that quickly can be minutes to hours, depending on concentrations and so forth. But there are a number of modifications that one can do that we implemented in our lab, and Ika Wamholk has a bioarchive on that as well, that range from pegylation to binding minor groove binders that inhibit DNase I activity, and what Ika sees is the ability to stabilize the particles out to 24 hours roughly. So that's nowhere near the 4 weeks that Daryl Irvin's lab has seen with Bill Schief for the EOD-GT8 protein nanoparticle that they've looked in germinal centers and seen, and lymph nodes, and seen that these particles can remain intact for 4 weeks, which, but at the same time, it's a very active area of research on how follicular dendritic cells actually recycle these intact particles and don't break them down and degrade them, and can also keep them intact. So, the in vitro DNase I and serum model or proxy is just a model for what happens in vivo, and we look forward to seeing, how long they're stable and how long we can stabilize them also during affinity maturation. Lauren had a great question. So about have you ever used a combination of different antigens on a single VLP, and do you think that strategy could increase the immunogenic response? And so the answer is yes, both. For our lab, we're pursuing that, and Aaron and Daniel have some excellent ideas about how to integrate multiple antigens from multiple coronaviruses into these nanoparticles to try to generate a broader immunogenicity from either past strains or potentially also future strains that may emerge. And then in the protein-based virus-like nanoparticles from Neil King that I showed as well, they also have two components, sort of mosaic particles, for example, for influenza, where they mix also multiple strains of HA in order to generate a more broad immunogenic response. Should I keep going, or?
Yeah, we don't have any others with the hands up. I'll
okay. So Jeff Moran asked, assuming that the DNA VLPs are shown to be efficacious in activating an immune response, what do you see as the main impediments to implementation of this technology at scale? And so that's a great question. I think the question of scale is always a really important one. Fortunately for vaccines, well, I should say vaccines are kind of an interesting case, because on the one hand, the doses compared to gene therapeutics that we see for AAV, for example, are minuscule. They're very, very low doses that are needed for vaccines. On the other hand, if you want to produce enough to vaccinate hundreds of millions of patients, or I should say people, or even 1 billion or more, then you get back to the challenging scale question, and that's in contrast to gene therapeutics for genetic disorders, where you might have a very, very small cohort of only 1,000 patients or 10,000 patients that need the treatment, but the dose per patient can be very, very large, and has led to what we've seen in the news as exorbitant costs of treatments per patient. So in our case, the scaffold that's produced is fully scalable to multi-thousand liter batch production of the single-stranded DNA scaffold that's produced in E. coli and phage. The single-stranded DNA is produced in our lab using phosphoramidite chemistry, or synthesis, I should say. And that's the same synthesis that's used for siRNA and ASO formulations for gene therapies. And so, somewhat scalable. There are other strategies published by, for example, Henrik Dies's lab and William Shee's labs, where they've biologically produced the staples as well by integrating them into phage's genome and then introducing restriction enzyme or DNazyme cutting sites, where you can then produce these staples also at scale. And so if you have the right sequences for hybridizing the antigen through its peptide nucleic acid or other hybridization motif, then that part of the formulation will scale also very straightforwardly. That leaves us with the anagenic protein, and of course protein production depends very much on the protein itself, how scalable it is, but generally for the types of anagenic proteins we're looking at, they're fully scalable as well. There are some conjugation reactions and purification reactions that should be industrializable. Overall, I would say very scalable. The question will be, of course, cost-wise. The goal of vaccines is not just to have an efficacious vaccine that gives us immunity for many, many years, in addition, obviously, to being safe, but also has a very low cost per dose, which some of the formulations that are currently entering phase 3 don't have yet. And so, that's a question of whether one can produce the vaccine, not just for the first world countries, but also for the developing world, where, costs have to be much lower to really impact the pandemic. So, overall, I don't have a direct answer to the question, but I think there are, a number of technologies in place now to, facilitate economically viable clinical scale production of these nanoparticles, assuming they're efficacious and safe. Let's see, so how do you see industry partnerships with your lab enabling rapid testing and deployment of VLPs for COVID-19? We were awarded a FAST Grants award from the FAST Grants Consortium that's been very helpful to kickstart the work with on our VLPs. We've also submitted a revision, one-year extension to an NIH R21 that we have that will hopefully be funded soon, which with the goal of that one-year catalytic funding from the NIH would be to actually translate these VLPs to commercial partners that could develop them. And we're working with local consultants in biotechnology to try to help figure out what the best path forward would be towards commercialization, assuming that we see positive results from Daniel's and Aaron's work over at the Ragon Institute in mouse models to be able to get to non-human primate models and the very expensive sort of ongoing studies that would follow that. So there was a question about antigen presenting cells and for cellular immunity, and can these particles be effectively processed, from David Gifford. It's another excellent question that I think Daryl and Daniel's labs are going to be able to, with Aaron, help us decipher. I think just one observation that's very important is that our particles consist of DNA plus antigen. Where the antigen is really targeted towards generating the neutralizing antibodies. The DNA itself, there are, as I mentioned before, endosomal sensors, TLR9 for DNA that, much like a viral genome, like adenovirus that has a DNA genome, trigger innate immunity, and there's a question of how that triggering of TLRs will either help or not help in the processing of the antigen displayed on the particles that then get processed and displayed to T cells for help through MHC2. And it's just an open question we just don't know the answer to. But if you compare it, that's one discrete difference between the protein nanoparticles, where you have a large amount of protein, megadalton scale protein, that's just the scaffold itself. And then the antigen of interest is part of that huge complex. And so you could argue that there might be a lot of distracting protein being processed in the protein-based virus nanoparticles that's not focusing the B cell repertoire on the antigen of interest in immunization, compared to our DNA nanoparticles, where we are only presenting the antigen of interest in the protein to the B cells to select those clones that are active towards that antigen of interest. So, but then again, the TLR9 signaling is kind of a question in the uptake. But there are ways of modulating the TLR9 signaling as well, as medicinal chemistry has been developed as well for siRNAs and ASOs extensively to sort of temper that innate immune response, or even eliminate it in some cases entirely. So we might need to look to those technologies as well. Guess we're at the hour, so thanks again, Justin. I think, do we wrap up at 1, or?
Yeah, we do. If you'd like to take any others, you certainly can, but we are at the end of the time, so
yeah.
Understand if people are dropping off.
I think I'll let everyone, I think we had a really good set of questions. They're great questions. Thanks everyone for them, and had a good discussion, and look forward to seeing everyone in person. We're hoping that all these vaccines work, and that we get to see everyone in the spring or latest summer in person, or if not sooner.
Great, and I'd like to, on behalf of the whole audience, thank you, Mark, for taking the time to tell us about this exciting work.
Thanks again.
And look forward to everybody joining us again in a couple of weeks as we have more installments of the series. So otherwise, I hope you have a good afternoon and the rest of your week.